Hi everybody, welcome to Chapter 7. This is going to be a review of neurons, and then we will have a review of the nervous system in Chapter 8. Okay, so specifically we're going to be talking about the cells of the nervous system, structure of neurons, how these neurons talk to each other via nerve impulses and synaptic transmission. Okay, so looking at the overall function of the nervous system, it's basically like the electricity that runs through your house. It's going to sort of control things that your body does, make different parts, different organ systems of your body talk to each other. There's two main divisions that it's divided into. You have the central nervous system, which includes the brain and the spinal cord, and then the peripheral nervous system, which is going to include nervous tissue outside of the CNS. This would include things like spinal nerves and cranial nerves. Nerves that go to your muscles or nerves that go to your face. So the divisions we'll specifically more talk about in Chapter 8. Now, let's specifically talk at the cellular level. We have two types of cells that we find in the nervous system. Neurons, these are the ones that transmit electrical impulses. They're also commonly known as action potentials. And so when you want your muscle to contract, it's a neuron that will send a change in voltage, almost like a lightning bolt stimulus, to that muscle cell in order to make it contract. Now, these neurons, they're amitotic, which means they don't divide. So what you get is what you get. And so they need some support. And the supportive cells are going to be called glial cells. An older term is neuroglial cells too as well. There's various types. Then has immune function. Some has nutrient function. So they provide support, growth, and also they produce what's called myelin. We'll talk about myelin in just a second. All right, there's three types of neurons. We're just going to go right to this picture right here. You have what's called sensory neurons, you have what's called interneurons, and you have what's called motor neurons. And so just looking at this image right here, a sensory neuron would be something that would detect sensations, touch, pressure, pain, temperature, and a sensory neuron can also be things like photoreceptors, like in your eyes, rods and cones, things that detect hearing, things that detect smell. So that sensory receptor is usually going to send information via the peripheral nervous system to the central nervous system. Sometimes it's directly within the central nervous system. And that sensory neuron is going to send information into the brain or spinal cord. It's going to interact with what's called a interneuron. Sorry about that. It's going to interact with what's called an interneuron. Interneurons are the most common neuron. They're only found in the brain and spinal cord, and that will then talk to a motor neuron, okay? Now, when these sensory neurons go into the brain, okay, the brain, that interneuron, that brain makes a decision about what it wants to do with that sensation. So if you look at this image right here, we're touching something hot. We obviously want to move our hand away. So the motor response would be a motor neuron, would be to contract that muscle to move that finger away. So if we're touching something with our finger and it's hot, that message will travel up our arm, into our spinal cord, and then up to the brain. The brain will make a decision. That motor output out of the brain will stimulate that muscle that's right here. So that is a motor neuron. All right, now let's look at the parts of a neuron. And we look at the parts of a neuron. I'm just going to go right here to this image here. The parts of the neuron that we have here, we have the nucleus, and then this is the cell body. Sometimes they call the cell body a soma. The cell body, it's typical for what we have of a typical cell. You're going to have organelles like Golgi apparatus, endoplasmic reticulum, etc. But attached to this cell body is going to be what's called dendrites. And dendrites receive information from our environment. So for example, when we were touching that heat, so when we say from the environment, that could be like touch, pressure, pain, etc. Or it can be from another neuron. For example, neuron A can talk to neuron B, which can talk to neuron C. And so the dendrites would be the receiving end. The dendrites would be the receiving end. They're going to be receiving that message from a previous neuron or some sort of environmental stimulus. Now, if that stimulus is strong enough, that action potential, that change, that electrical impulse, which is actually just a change in millivolts, will travel through the cell body and it'll travel along what's called an axon. And so this axon will conduct the nerve impulse. And look at here, we have another neuron. So this is, let's say this is neuron A and this is neuron B. And at the end of the neuron, we would secrete chemical messengers to stimulate this neuron. Now, sometimes the message doesn't completely go from neuron A to neuron B. You need what's called in between here, we'll talk about this later, you need what's called neurotransmitters. And these neurotransmitters, these are chemicals that basically cause the nerve impulse to travel from one neuron to the next neuron. Okay, now, looking at here, we're talking about nerves. So nerves consist, like nerves that we think about that are running through our muscles. Those are bundles of axons. They do have dendrites too as well. They're usually surrounded by connective tissue, and they can either be sensory or motor, or both sensory or motor. So it really depends on the type of nerve. We'll see nerves in the next lecture a little bit closer. All right, so myelin. So myelin is this lipid-like sheath, this lipid-like protective layer that actually increases the rate of an action potential. And I'm just going to go right to this image right here. So here is our axon right here, this yellow sort of string-looking like thing. And then here is this wrapping of this lipid sheath called the myelin sheath. And depending on which cell you are, Schwann cells, they produce this myelin in the peripheral nervous system. So think about like nerves that are in our like arms and legs. And then another type of cell will produce it in the central nervous system called an oligodendrocyte. But the whole idea with myelin is it speeds up electrical impulses. And what this is showing is the impulse is jumping. It's jumping. This is called saltatory conduction, and it's jumping from these little nodes right here. These nodes are called nodes of Ranvier. And so instead of the impulse traveling through all aspects of the axon, it jumps. It jumps. It's kind of like what's easier to do, either skip rocks across a river or swim and tread through the river. It's much easier to skip across. So myelin, it allows us to conduct impulses a lot faster. There is a disorder of myelin called multiple sclerosis. We commonly call it MS. And so it's not really known what causes it, so they use the term idiopathic, but there's some sort of attack. There's some sort of attack against the myelin sheaths. And so they think it's like maybe some sort of autoimmune, but either way, it basically causes scar tissue to occur in this myelin and therefore impairs with the electrical signal traveling to where it needs to go. So we can get altered sensations, and sometimes we may not be able to control our muscles too as well. Okay, so let's go ahead and talk about this nerve impulse. So nerve impulse is kind of your day-to-day term, but the more scientific term for it is an action potential. And an action potential is really a change in voltage across a cell membrane. Now, what causes this change in voltage? This change in voltage is caused by the interaction of sodium potassium crossing through the cell membrane. You guys remember the plasma membrane. Now, they pass through what's called ion channels. And so I'm going to show you a picture right here, okay? And so this is looking at a cell membrane. And before we look at this picture right here, I'm just going to draw a membrane off to the side. Here was a membrane, a cell membrane, and this is inside and this is outside. Now, sodium likes to live, Na stands for sodium, sodium likes to live outside the cell. Potassium likes to live inside the cell. And usually the cell, when it's resting, meaning that it doesn't have a charge, you're going to see a negative charge on the inside of the membrane and a positive charge. And you're probably like, wait a minute, I see a K+. So why is there a negative charge? There's also proteins in here too as well. And these proteins, they carry a negative charge. Now look at right here. We see resting neuron. You can see this description right here. Now, something is going to cause sodium to come into the cell. And usually there is what's called a neurotransmitter. I had mentioned that previously. A neurotransmitter, which is a chemical messenger, will open up an ion channel. I'm just going to make a channel right here. And that will cause sodium to rush into the cell. Now, as sodium rushes into the cell, you can see the inside of the cell become positive. So as sodium is rushing in, we're kind of looking at this right here. Look at the positives. You can see that the inside of the membrane is becoming positive. This term right here is called depolarization. So depolarization, and the way that they describe it is depolarization, the inside, and it's really just on the inside of the cell membrane, the inside of the cell membrane is becoming less negative. This is your action potential, and this is going to roll down the entire membrane, kind of like knocking a row of dominoes down. Now, as this is because sodium is coming in, this is becoming positive, what's going to happen is we need to restore that inside of the cell to its negative roots, and so what's going to happen is potassium's going to leave. And so you can see potassium leaving right here, and that's restoring the negative value right there. And so that is called repolarization. And repolarization is the restoration, the restoring of the resting membrane, and I'm going to put negative value, the negative value inside. Now the problem that you'll have here is when we look at this closer, now we have sodium in here and now we have potassium out here. At the end, what will fix this, so with potassium leaving, we're going to restore that negative value, but what fixes these ions to where they need to go is going to be a sodium-potassium pump. So a sodium-potassium pump will restore sodium outside and potassium inside. Okay, so this is just showing you guys here some numbers right here. So the inside of the membrane, I said that was negative. It's usually sitting at negative 70 millivolts, okay? And so here we're looking at resting. So we're at negative 70, negative 70. At this point in time, something lets in sodium. You can see this increase in value or becoming less negative. If you look at this, this is 0. So sodium is coming into the cell. This is all represented by this uphill, going up the hill here. And so you can see we get all the way up to positive 30. At positive 30, right at this point here, no more sodium comes in. No more sodium in. And potassium leaves out. Okay, so no more sodium coming in, potassium leaves out. So this part right here with the sodium coming in, this is your depolarization. This is your depolarization. I'm going to clean this up here. So this part right here, this is this sort of lighter brown here. This is your depolarization. So at positive 30, no more sodium comes in and potassium channels open, and this is called your repolarization. Your repolarization, okay? And then after this, your sodium-potassium pump will fix your ions. And so if I just draw a membrane right here, sodium, remember, likes to live outside, potassium likes to live in. So something lets sodium come in. When the inside of the membrane hits positive 30, these membranes, these ion channels shut down and potassium starts to leave. And so that's great. You know, we restore it back to negative, but we've got sodium inside and potassium outside. And so that sodium-potassium pump, they'll fix it by pumping sodium out and bringing that potassium back in. All right, so once you get an action potential going, once it hits what's called threshold, there's really no stopping that action potential from happening. It's an all or nothing effect. There is a portion in time called the refractory period, and so this is in this area right in this region right here where you basically cannot get another action potential. It's really when sodiums are entering into the inside of the cell there. Okay, so when we talked about one neuron talking to another neuron, this is called synaptic transmission. And so it does it by what's called neurotransmitters. And neurotransmitters are basically chemical messengers that occur between neuron to neuron, or maybe a neuron talking to a muscle cell. A synapse is when you have one neuron talking to another cell, which could be another neuron or a muscle cell. And so let's just go ahead and look at this image right here. We have what's called the synaptic bulb right here, and we have vesicles. And in these vesicles, we have neurotransmitters in those vesicles. And so you usually get some sort of message coming down, another action potential coming down. That will cause, let me write this again, you have another action potential coming down. The axon would be up here. This synaptic bob is also called the axon terminal. And so this action potential is a nerve impulse. It comes down, okay? It causes these vesicles to fuse with the membrane and dump into what's called the synaptic cleft. So the synaptic cleft is sort of this region right here. And so with these neurotransmitters, when they attach onto their receptors on the next cell, so this is a cell, it could be another neuron or it could be a muscle cell, what will happen is it will cause the exchange of ions. It will cause the exchange of ions. And so we have sodium coming in, and then you would have maybe potassium going out and causing another action potential in this cell here. It really depends on the job of what the cell is. Okay, this is talking about how calcium ions, when they come in, if you guys see right here, when you have your action potential coming down, this is the next slide is specifically talking about when this action potential comes in, calcium comes in. Let me just restate that. The action potential comes down, calcium then comes in. You can see these calcium channels right here, and it's specifically the calcium coming in that causes these vesicles filled with that neurotransmitter. It causes them to fuse with the membrane and dump into what's called the synaptic cleft. Okay, now this neurotransmitter that we have here, this neurotransmitter, it can either be excitatory or it can be inhibitory. So it really depends on the chemical that's in there. Types of neurotransmitters that we have is acetylcholine, serotonin is another type. Obviously, an excitatory would cause the next cell to likely have another action potential, whereas if you were inhibitory, that would usually prevent that next cell from having an action potential. And so another thing too to think about is we may have many more synapses on these neurons here. And so looking right here, you can see this is one cell body here. It has excitatory in green and it has inhibitory. So sometimes you can have combined effects of excitatory and inhibitory, which can cancel each other out, or you could have more excitatory, or you can have more inhibitory. This is called summation. All right, and that is it. You do have a couple other disorders that they talk about in your lecture, so make sure you guys read up on those, on different types of neurotransmitters that exist at the end of your lecture notes. Thank you.